welcome, and first of all, thanks to the organizers for inviting me here. I am assistant professor from University of Silesia, and I'm working with this smoldering always damps environment. Uh, and the talk I'm giving today is about a sort of side project that came out. So, what are the smoldering dams? So, always dams is a pile that is formed from the waste material after mining activity, and it's rich in some in coal and organic compounds, as well as some heavy metals, other minerals. It's generally pretty nasty environment to live, especially where if there is a lot of pyrite, it can interact with water and oxygen, which causes oxidation that generates heat, and if it's inside the pile, then this heat increase quite a lot and cause the organic matter to start burning, and we end up with something like this. Uh, Here you can see one of the example of the colorless dams which we sampled. It was taken a couple years ago in November, so there was ambient temperature was surrounding temperature was about 5 degrees above zero, but the areas on the colorless damp can reach at the surface between 4,400 Celsius degrees or even more. There are also some quite pH extremes, and most of it is very acidic. However, there are some alkaline spots. And here you can see some example how diverse these environments are. There are some salt precipitates, there is some bitumen leaking, uh, sulfur precipitate, as well as some different environment after the organic matter is burned out. Um, yeah, just to show how bad this environment is for living organisms, here are some analyses of heavy metal content, and there is quite a lot of mercury, which is, as we know, very toxic. There is some significant amount of zinc, lead, and also some nasty organic compounds, including haloorganics and polyaromatic hydrocarbons, as you can see. Here there is quite high percentage of those. 2-3 rings in blue, 4 rings polyaromatic hydrocarbons in green, and 5-6 rings polyaromatic hydrocarbons in yellow. And those are data from 2 dams that we work on. The main project I'm working with was focused on the metagenomic analysis of these spoils. However, about a year ago, a group of students came to me that they said, we have a lot of free time and we want to spend it in the lab and work for you for free. And then I came up with an experiment that actually I was thinking for some time, and it was inspired by a paper by Sugar et al. From 2013, when they took microorganisms from soil and tested them in some polyextreme conditions where they changed their atmosphere to CO2 atmosphere. They incubated them at 0 degree and in very low pressure. And what they have seen that out of almost 30 microbes they tested, one was still able to grow in these conditions. And this was Ceratolipiphaciens. In the follow-up studies, they look at the transcriptomics of these microorganisms growing in this condition, and they could see a number of genes associated with different pathways that are both upregulated and downregulated. Here's an example of upregulated genes that were connected with some sugar metabolism, with amino acid synthesis, with ribosome processing, or amino acid tRNA biosynthesis. However, we couldn't afford to do the transcriptomics, so I was thinking what we can do except only testing if the microbes can grow in such conditions. Then I thought about one of the most crucial structure in living organisms, which is the cell membrane. And especially for microorganisms, it's crucial because it also contains the energy conservation machinery. And the cell membrane, for those who don't know, is formed from aliphatic molecules that form the bilayer. and is the barrier between microorganisms and environment. And in microbes, we distinguish two main types of these membranes, like in gram-negative bacteria, there is just a plasma membrane and thick um, cell wall. However, in gram-negative bacteria, there is plasma membrane, a thin cell wall, and then also outer membrane. And cell membrane, in order to perform its function, has to have certain level of fluidicity, and this is maintained by changing its composition. It can have be more rigid when there is more saturated fatty acids, or more fluid when there is more unsaturated fatty acids. There can also be hydro fatty acids, fatty acids, and so on. So in general, in high temperature, the portion of saturated fatty acids is increasing. In the low temperature, saturated are reducing because they make the membrane more fluid. So the aim of our study was to examine the cell membrane composition uh, and its changes when those microorganisms from these thermophilic polyextreme conditions are exposed to uh, growing conditions that are quite distinct from their native environment. So, like I said, we had isolated some microorganisms. We obtained the pure cultures, then characterized them by both morphology and molecular approach, and then we grew them or aimed to grow them in several different conditions, which involve changes of atmosphere between our normal N2O2 atmosphere and CO2 rich atmosphere, different temperatures of incubation and different pressures. And after that, we also going to do analysis of morphology and this fatty acids composition. In order to do that, we had to build our own DY planetary simulation chamber, again inspired by this built in the by Schruger et al., which was composed of a vacuum pump connected to excicator that can withstand the total vacuum. And here are some examples of results from different conditions, and you can already spot that the condition influenced the microbes. These strains that Serratia strains that were producing this dye, they lost the coloration. Some of them grow better, some of them grew worse in those different conditions. Um, Here is we all over we tested about 40 strains, and here are examples of our thermophilic strains. However, they grew most of them pretty well. We lost one in our lab at 70 degrees, but already at 25 degrees they couldn't grow at all, and the same was for 25 degrees and low pressure. For sorry, for mesophilic strains, we got better results. So besides those from colonizers, we also included some strains that came from mines, coal mines, and we included Bacillus subtilis and E. coli as the controls. And you can see that the temperature was affecting them the most. However, from all of them, we choose two that that showed the best active growth. So we didn't only look at the survival of microorganisms, but also at the active growth in those conditions. And there was one strain, this over here, which seems to be better choice for further studies. However, we figured out that it was still mixed strain. So we right now are trying to separate the two strains. There is Serratia marchescens and species of Dixonia. And so, like I said, we choose these two strains for follow-up studies, and I will show results for one of them. Basically, it's easy to be free. So here is gram strains is short rod-shaped gram-negative bacteria, and 16 phylogenic analysis show us that is closely related to Xenophonus alcophylla. However, I wish it been used, although we don't have enough data at this time point to be sure for that. So now we add its fatty acids composition. This is the overview in the what we called ambient conditions, 25 degrees, normal atmosphere by pressure as 1%. Majority of the fatty acids in the membrane were saturated ones, and there was a small fraction of unsaturated. It wasn't surprising that at low temperature, the unsaturated fraction increased significantly, while the saturated fraction went down because, yeah, in order to maintain higher fluidity. But little bit to us that at low pressure, the unsaturated fraction goes up while the saturated went down. When you look on the saturated fatty acids divided by different hydroxide oxidated fatty acids in this blue color, we can see that these two classes of branches cyclofatty acids are completely gone at 4 degrees, and branches fatty acids is increased at the low pressure against the higher fluidity of the membrane. And number-wise fatty acids, and there are also changes increase of this 15 carbon branded fatty acids, while there is decrease of 16 carbon fatty acids, as well as increase of 7 or 16 carbon with unsaturated bonds fatty acids. When you change it to 4 degrees, we can pretty much same level as in the ambient condition. Why this fraction of 18 carbon containing fatty acids is significant.
Sorry, Piotr, can I interrupt you for a moment?
And
do you hear me, Piotr?
Also, kind of surprising was that the two types of positive fatty acids significantly increased for this particular strain. So the conclusions we draw so far is that temperature can be to be the major factor influencing growth. 38 of the strains that we 8% of the strains that we tested were able to grow in these conditions, and it seems that gram
Excuse me, Piotr, do you hear me? Can I interrupt you for a moment?
But we can.
We have the problem with the connection. Your voice is still not clearly heard by us. Do you have any problems with the internet connection?
Okay.
We can see the presentation, but your voice is quite difficult to be heard.
No, it seems fine on my side.
Okay, so can you continue?
Okay,
because a few persons told me that there's a problem with the internet, probably. Okay, so continue.
Yes. So so we could see that there are some changes in fatty acids composition upon these different conditions. However, we do not draw final conclusions yet because the project is still ongoing and we weren't able to test the gas. The most important conditions were both low temperature and low pressure are applied. The reason for that is because our vacuum and then this is all, so we cannot really service it, so we're looking for some round solution. So this is something for the future that we definitely will test, hopefully in the near future. We also think it will be interesting as well as to examine the growth, microbial growth in the presence of other stressors like, for example, perchlorates or maybe radiation. So this is also our, and I would like to thank especially Anna, she's doing good
Excuse me, Piotr, could you
and so receive this the best grant from Institute of Education and Science.
excuse me, Piotr, do you hear me? Can you
helping
turn off your camera, because we will be able to hear you better. I think that is a problem with the connection of internet too low. Oh yeah, thanks.
All right.
That's. Can you?
It should be off now.
Can you tell something else now?
Mm-hmm.
Okay, I think it could be better. It could be better
yes,
without the camera. I think that yes, here we can talk, so we'll see. Okay. So thank you very much for your presentation.
Okay. So yeah. Yes, and
we can't hear you now.
So thank you, and here are some contact information